Bonjour. Can I ask that you can take your seats, please, for the session? Bonjour. Welcome and thank you everyone. If you can take your seat, we will start the fish spawning aggregation session for GCFI77. Woo! So thank you for all attending this session. Fish spawning aggregations are a very unique thing, and I can say that the amount of people that work on spawning aggregations globally is quite small. And so it's interesting that because spawning aggregations are, I think, a very, very important thing that a lot of reef fish species actually gather to spawn. And we know some things about it, but I think there's a lot of things we don't know about fish spawning aggregations and why. Why they gather, where they gather, and when. And so this session is a session we have at GCFI every year to gather and talk about all the aspects of fish spawning aggregations. So we have five speakers today. The first speaker is a virtual speaker, and let's just get straight into it. The talks are 15 minutes, but that includes questions. And so I'll try to keep to time. So to kick off the first presentation, we have Kyla Binkau, virtual presentation. Do we have Kyla ready? To talk to us about yellowtail parrotfish. Welcome, Kyla.
Good day. Today we'll be sharing some of the research that we've been doing on yellowtail parrotfish in the U.S. Virgin Islands. All of this work is a part of a larger Virgin Islands EPSPOR and SF grant that is titled Ridge to Reef. So since 2020, we've been monitoring yellowtail parrotfish populations in Reef Bay on St. John. This is a picture of beautiful Reef Bay. This is a map, a less beautiful map of Reef Bay. And we chose Reef Bay as the site of this study for a couple of different reasons. First off, there's evidence of a sedimentation gradient in this bay. So the prevailing wind patterns kind of throughout the year are from east to west, and this causes kind of a loading of sediment on the western side of the bay. In addition, there is increased development on the western side of the bay because a portion of the land area on that side is outside of the national park that takes up most of St. John. So we have increased development and increased opportunity for runoff on the western side of the bay. And we suspect, and there's evidence, that this results in increased sedimentation on that western side as compared to the eastern side. The second reason we chose to use Reef Bay for this study is that it's home to 2 yellowtail parrotfish spawning aggregations. And for those of you who might not know, yellowtail parrotfish are weirdos. In the initial phase, males and females actually undergo almost daily migrations to their spawning aggregation sites. And not only that, but they show a shocking level of site fidelity to their aggregations. So in Reef Bay, we actually have an aggregation that's located on the west end, and all of the fish that live on the west side of the bay go to that aggregation. And then we have another aggregation on the east end of the bay, and that's where all the fish that live on the east side of the bay go to spawn every day. And so these aggregation sites are shown on the map by that circle with the X through it. And this spawning behavior essentially sets up a natural experiment, allowing us to compare spawning activity between the east and the west to understand how different environmental factors on different sides of the bay influence the spawning of these fish. So since we're all fish nerds, I figured I'd treat you to a video of some spawning from the aggregation on the eastern side of Reef Bay. So we just saw a spawning rush. And another one. You'll see a couple more throughout this 30-second video. So incredibly, these aggregations form almost daily. So every single day, these fish are undergoing a migration to the aggregation sites, and they are spawning and performing these spawning rushes. So this is offering us to gather a lot of data about their spawning behavior. One of the main ways we've been monitoring these fish is by using acoustic telemetry. So we tagged 60 individuals from 6 different regions of the bay using acoustic transmitters and tracked their movements for a little over a year. In addition to tracking fish using the receivers that are denoted by the black dots on this map, we've also been monitoring water temperature, sedimentation rates, the benthic communities, and fish behavior at their home reef sites. So today I'm going to share a little bit of what we've been finding. This is a really big project, and there's a lot of results that we're just kind of brushing the surface of now and sifting through to kind of get a full picture of what's going on. So I want to start by sharing some results from Jenna Sheremy's master's thesis. She was a master's student working with Rick Nemeth, who's the PI on this project. And she found some really important things related to our yellowtail. First off, she was able to confirm that there was a sedimentation gradient across the bay, and it behaved as we expected. So there was more sediment on the western side of the bay compared to the eastern side. She also found 100% site fidelity of fish to their respective aggregations. So all of the fish that were tagged on the east side of the bay went to the eastern aggregation. All the fish that were tagged on the west side went to the western aggregation. When looking at the tagging data and looking at visitation frequency to the aggregation sites, Jenna found that the western fish visited their aggregation more frequently, but they spent less time there. She also was monitoring foraging rates and found that there were lower foraging rates on the western side that had higher sedimentation. So at this point, we're not really sure why we saw the results that we did, especially regarding the aggregation visitation frequency and duration. But we suspect that this might have something to do with the lower quality forage available on the western side where there's increased sedimentation, which has been shown to influence reproductive output. And it could potentially be the result of differences in sex ratios. So we're working with some collaborators to kind of tease apart some of these things, and I'll talk about that more at the end of this presentation. So from Jenna's thesis, we learned a lot about how these parrotfish are interacting with their aggregations, but there's still this gap regarding what the tagged fish are doing while they're at their home reef sites. So we're interested in asking the question, how does activity differ when fish are at their home reefs and during the migration itself? So I'm going to be sharing some of those results here. And what I'm showing you here is the kernel utilization distributions for our fish, specifically outside of the spawning time window. And so you're basically seeing a repeat of the Reef Bay map for each of the different 6 locations where we tagged fish. And what you're seeing in the colored areas is the 50% kernel utilization distribution, and then the grayed out areas is the 95%. And what I'm gonna be referring to the 50% KUDs or kernel utilization distributions as the core use areas, and that's where I've kind of focused this analysis, because this is where I suspect or this is where we know that the fish are spending the majority of their time when they're at their home reefs. So on average, we saw like very similar core use areas between east and west, but notably there was a lot more variability in the size of core use areas on the eastern side of the bay. Also, potentially interesting, I'm not really sure what this means yet, we found that almost all of the fish hung out right where we dropped them off after tagging. So I made these same plots including a point where their release location was, and almost every single fish had a core use area that surrounded its release location. We're also interested in seeing whether there was a sequential nature to the migration timing of the fish from the different areas of the bay, thinking that perhaps the fish that live on the inner parts of the bay would migrate earlier and cue the others to migrate as well. And so these plots are showing you the probability of each individual fish departing its core use area to undergo its spawning migration by the time of day. And what we found was that there was a sequential migration in the west, with fish with core use areas kind of in the inner part of the bay leaving earlier to start their migrations. This relationship didn't hold on the eastern side of the bay. But I suspect this could be due to the small sample size associated with the fish from the outer edge of the eastern side. So I don't necessarily know if this is something that's actually naturally occurring or just an artifact of that small sample size. So we were also interested in seeing if there were differences in the timing of spawning. And so this plot is showing the probability density of fish being detected at their spawning aggregation receivers by hour of day. And the red is showing you the eastern aggregation fish, and the west is showing the western aggregation. And so you can see that the western fish peak earlier. They're most likely to be detected at the aggregations around 4 p.m., while the eastern fish peak between 4:30 and 5 p.m. And we're not really sure what causes this difference. It could be due to differences in oceanographic patterns between the two sites being differently optimal for spawning. There could also be differences in whatever cues the fish to migrate in the first place, or it could just be the fact that the eastern side of the bay is longer than the western side, and so the fish are just taking longer to get there. So combining our understanding of fish movements at their home reef sites and their spawning areas, we've been able to document that there are clear differences in the spawning behavior between the eastern and western side of Reef Bay, but we're still not very clear on why these differences exist. And at the moment, we're kind of finalizing these analyses and incorporating additional data that will allow us to kind of tease this apart. And so some of the data that we're starting to incorporate includes those environmental covariates, including temperature, as well as some weather data and current data that we've been collecting at the aggregation sites. We've also sampled yellowtail parrotfish as well. And we are looking at their gonatosomatic index to see if there's differences in that between the aggregations. We're also looking at gut contents to see what they've been foraging on, as well as the amount of sediment that they're consuming while foraging. And then we're also looking at direct measures of spawning frequency using sound and video. And I just want to close by plugging this really cool thing that my graduate student, Stephanie Maxon, has been able to uncover. So you might have noticed I said we're looking at spawning frequency by sound. And what's really amazing is that Stephanie has been able to isolate and identify a sound associated with the yellowtail spawning rush. So when the yellowtail, the female goes up and flicks back down really quickly, that flicking back down actually makes a sound. And Stephanie has been able to consistently identify that sound, and we're looking into using passive acoustic monitoring to quantify the number of spawning rushes that occur at each spawning aggregation. And we're hoping that'll give us a better idea of differences in the spawning output of the different aggregations within the bay. So with that, I'd like to open it up for questions and just thank specifically our funding source, the National Science Foundation BIF Score project. And thank you for listening.
Thank you, Kyla. That was very interesting. Did I hear, is Kyla actually there? Yes. Kyla, did I hear you say right at the beginning that yellowtail parrotfish are weirdos?
Yeah, I did say they're weirdos.
Excellent. Just what I would like to hear. Any questions? We've got time for a question.
Hi. I have the mic, if that's okay.
Oh, Terry, of course.
Mike gets to ask the question first. Terry Donaldson from the University of Guam. Very interesting talk. I have a two-part question, and I promise I'll be brief. First, do you have any evidence for differences, significant differences, between abundance of participants in the two aggregation sites? In other words, one site is equivalent to the other in abundance, or one site is less versus the other site, or more, as the case may be.
Do you want me to answer that question first, or do you want to
yeah, it's kind
have
of
your second part also?
It's predicated on your cheerful answer. Yes, please. Yeah.
Okay. So we have been monitoring with video, and so we have been looking at like max N at the spawning aggregation sites to look at the abundances. We really haven't seen that much of a difference in terms of the number of fish that are migrating to the aggregation. That being said, we've had a really hard time with our video monitoring. I think chiefly because we're having a hard time with our battery power on our cameras, and so we really have to be there to deploy them and have them running while that peak spawning period is happening. And sometimes we capture it and sometimes we don't. And so I'm not super confident at this point to say whether we think, you know, one has more or another. But we have also been doing some monitoring as well on reef breathers, because the fish do respond to us when we're down diving, observing them. And so we do fish counts when we're there. But like, definitely they respond to our bubbles and us being there, and their behavior is different when we observe them in water compared to when we're observing them on the video. But they tend to have less of a response when our rebreather divers are down. So that's another thing we're kind of looking into doing more, is getting more fish counts with our folks that are able to go down on rebreathers.
Okay, that's good. So this prompts the second part of the question. Parrotfishes are notorious for having alternative mating strategies, all right? And this can be brought into play based on abundance of individuals at the aggregation site, and also, ironically, the type of benthic structure you have at the bottom. Is it you know, wide open? Are there landmarks versus is it cluttered full of sediment, as you've shown it can be? I noticed from your video you've had a lot of group spawning. Have you seen any other types of spawning events, that, like pair spawning, for instance, males setting up a territory, temporary territory, and females going to that male at the aggregation site?
At one of our aggregations on the western aggregation, we do have video of a male. And we don't know if that male is like resident there and that's where its territory is, but he tends to get really annoyed when all the female or all the initial phases are there spawning, and like he gets really agitated and kind of chases everybody around. I don't think we've ever actually observed pair spawning among yellowtail at our aggregation sites. We have observed spawning of other species just from our video monitoring as well. Like these areas, I think, are particularly on the western side, but also on the eastern side, like they're kind of out on the reef edge, and they both have like relatively diverse aggregations, like relatively diverse groups of fish living there, and we have observed spawning of other species, but I don't think we've ever observed anything from the yellowtail parrotfish other than the group spawning that I showed.
Okay, great, thanks.
Thanks,
Please
Terry.
Tell Rick Terry says hello. Okay. Thanks.
Thanks, Kyla. We have time for just one more question over here. Will, a quick question, please.
How did you find the aggregation?
She wouldn't hear you. You need to use the mic, Will.
The question from Will is, how did you find the aggregation?
I did not find it. So, I mean, basically, and Sean Caddison is there in the audience. You guys can ask her this story, but I came on the scene after the aggregations were originally found. But I think one of the aggregations had been located, and there was another master's student many years ago, not that many years ago, but previous to Jenna Sheremy, who I talked about, who had been doing tagging on the aggregation that they had already identified. I think it was the western one. And like half the fish that she tagged in Reef Bay never showed up at the aggregation. And so Rick and Sean were like, okay, we think that there's probably another aggregation. And just thinking about the benthic structure and the way that Reef Bay is laid out, they kind of started to explore the area to look for where this other aggregation might be, as well as using the telemetry data from that preliminary master's thesis, and we're able to identify where that aggregation was. But I don't know if Sean's in the room and she wants to tell that story. If you want to follow up with her afterwards, she definitely knows more about the actual finding of the aggregation than I do.
Thanks, Kyla. And Sean is in the room, and she's going to be talking later as well. So I'm just going to move the session along. Further questions can be followed up afterwards, if you like. Thanks, Kyla.
Thank you.
Our next presenter is Janelle Layton. Janelle, welcome. Janelle is going to tell us all about tiger grouper and Nassau grouper.
Hi. Good morning. My name is Janelle Layton, and I am a current graduate student at Oregon State University, partnering with various organizations. And I look at understanding energy output during spawning aggregation events of Nassau and tiger grouper on Little Cayman Islands. Okay. So to start, energy is super important. It's the currency of the ocean. Studies as far back as 1942 have said everything in community dynamics is driven by energy. And we see that especially in reef systems especially in aggregating systems where eggs and larvae are released, where gametes are released and become a food source for smaller organisms. Some of those gametes then become larvae and become a food source for other organisms as well. Some of those larvae turn into juveniles and adults, and they become consumers of other organisms, and some of those larvae don't make it, and a lot of that energy gets transferred back into the ocean as nutrients. And so this energy transfer is extremely important, especially in coral reef ecosystems as a whole. Nassau grouper are distributed throughout the western Atlantic Ocean, yet they are listed as threatened under the Endangered Species Act and critically endangered by the IUCN. Major threats include things like overharvest and lack of effective regulations, but they're an important species ecologically as large reef predators, economically in a commercial and recreational fishery, and culturally. In dive tourism and other tourism culture, it's really important. They're a food fish, and in the Cayman Islands at least, they are incorporated into education systems and curriculums as well. Tiger grouper, also common throughout the tropical West Atlantic and an important large reef predator, but they are listed as data deficient on the IUCN Red List. We just know a lot less about this species compared to Nassau. So this is the spawning aggregation site that I study on the west end of Little Cayman and the Cayman Islands. On the right, you will see Nassau grouper spawning, and on the left is a video of a smaller, much smaller tiger grouper spawning site. They're gonna pop up in the back in a second in this video. And this is all happening, like I said, on the west end of Little Cayman in the winter months around the full moons. This is a shared aggregation site. Not only do tiger and Nassau spawn here, but also yellowfin, black grouper, and various other species have been noted here. And tiger grouper will generally begin spawning a few days after Nassau grouper begin at this site. So various conservation efforts exist globally, but I really want to focus on the efforts placed in the Cayman Islands. Things like gear restrictions, catch limits, closed fishing seasons, etc., has led to one of the biggest fish conservation stories in the Caribbean. This work has led to the recovery of Nassau grouper in the Cayman Islands, and this is a project called the Grouper Moon Project, which is a collaborative effort between the Cayman Island Department of Environment, Reef, a nonprofit in South Florida, Oregon State University, and Scripps Institute of Oceanography. So we know the importance of energy, we know the importance of these fish species, yet little has been done to actually quantify energy output during spawning of these species. And so this leads into a few research questions that I have on, does energy output vary across spawning night of specifically Nassau grouper? Does energy output vary between species, being Nassau and tiger? How much energy is produced in a single spawning event? And then how much energy is produced in a spawning year, and how has it changed over time? I already mentioned my study site on the west end of Little Cayman, and this is the fish spawning, and a team of divers will be out doing various things, but for my project specifically, a diver will be swimming around with a plankton tow and collecting as many embryos as possible throughout the water column, not really targeting any specific spawning event. And I end up in a lab with literally thousands and thousands of these embryos. This is what they look like under a microscope, and then this is what they look like to the naked eye. At this point, I'm counting a set weight of embryos to get a wet weight ratio for one single egg. And then those eggs get dried overnight and then turned into these small pellets that get weighed and go through a bomb calorimetry process. And the bomb calorimetry basically measures the heat release during the combustion of the single pellet. And that gives us the caloric content of a pellet. And this was done for 2 nights of Nassau grouper spawning in 2023 and 1 night of tiger grouper spawning in 2023. So back to my research questions. Does energy output vary across spawning nights and between species? And this figure is showing energy in kilojoules on the y-axis for one single embryo, and on the x-axis, different nights of spawning. The red is showing night 1 of Nassau grouper spawning, the green is showing night 2 of Nassau grouper spawning, and the blue is showing tiger grouper spawning. So we did see a significant difference between night 1 and tiger grouper spawning. So that is one thing that was expected because tiger grouper eggs are a little bit smaller than Nassau grouper eggs are. And then one thing that was also interesting was a slight difference between night 1 and night 2. It wasn't statistically significant between spawning nights, but we did see a slight decrease over time. And so unless you know what 0.013 kilojoules really means, I made some conversions to make some of this energy data make sense. The average human consumes about 2,000 calories a day, or 8,300 kilojoules a day. So it would take about 619,000 embryos to feed the average human for a single day on night 1 of spawning, which seems like a lot, but how many eggs are females producing when spawning? And so that goes into my next set of research questions about how much energy is produced in a spawning event and then a spawning year. And so to do this, I had to get average batch fecundity estimates, and that data was not available to me at the time, but another study, Collins et al. 2002, had done a study on red grouper where they could use length estimates to measure batch fecundity estimates, and I already had length data from the Grouper Moon project going as far back as 2003. So using that data specifically in the year 2023, we were able to get length estimates for 673 fish, and from that we could get fecundity estimates for an average fecundity estimate for one female in 2023. And in night 1 spawning, there was a little bit over 4,000 kilojoules of energy, and in night 2, around 3,800 kilojoules of energy released from just one single female. That's about feeding a human for half a day. So that's really cool for one female, but how many fish are actually in the water? And so that's when I got population estimates, once again, from the Grouper Moon project. They've been doing this project for well over 20 years, and I have data from the years 2008 up until 2023 on population estimates. And then hormone analyses from a while ago were able to inform me that it's about a 50-50 sex ratio for Nassau grouper at these spawning aggregation sites. So every value was basically cut in half to get the number of females in the water. Okay, so then looking at energy in a spawning year, specifically in 2023, we noted about 17 million kilojoules of energy being released on night 1 of spawning, and then a little bit around under 16 million kilojoules of energy on night 2 of spawning. That could feed a human for up to 5.5 years on night 1 and 5.2 years for night 2. It's a lot of energy. But how has it changed over time? Our aggregations weren't always looking this large. In 2008, it was much smaller compared to 2023. And while increasing population size may seem like that leads to increasing energy, that's not necessarily a direct relationship. Another thing to note is that fish size or length of fish impacts fecundity and therefore can also impact energy output. So having a lot of big fish could look very different than having a lot of little fish. And that's one thing to take into account when understanding this energy data. The lowest year was in 2010, where you couldn't even get a year's worth of energy to feed a human, whereas in 2021 was the highest with almost 6 years of energy to feed a human. A very dramatic difference when you have a healthy spawning aggregation site. So overall, energy is very important. A healthy aggregation can be contributors of up to 18 million kilojoules to an ecosystem that can feed a human for 6 years, which is really crazy. Energy dynamics can change with changing size of FSAs, but also length of fish. Allocation of energy can vary depending on spawning night, but also depending on species, so there's not a one-size-fits-all answer. You have to look at various things. And in future work, I would like to incorporate some of the 2024 data we just received, repeat the study to see if spawning night differences are consistent or if that was just in the year 2023. Add an additional spawning night for tiger grouper and also additional species. This is a multi-species aggregation site. Yellowfin grouper is another commonly studied species in this area. And then apply this work to a more of an ecological context. So this is only a slight part of my graduate work, but things like understanding energy output affecting predator-prey dynamics, nutrient availability, or should we be looking more at a bottom-up control of large reef predators rather than top-down when they're spawning? I'd like to thank all these people and GCFI for the travel award to come out here and present this work. Any questions?
Thank you, Janelle. I find the energy transfer story in spawning aggregations fascinating about if spawning aggregations are reduced, it doesn't only affect that species, it can affect a whole range of other species and ecosystem functions. So yeah, it's fascinating. We have time for a couple of questions.
Yeah, and I've already got the mic, Martin.
Where?
That was really nice, Janelle.
Thanks,
so
Nancy.
do you know if the same female is spawning 2 nights in a row, and if that would maybe account for the lesser energy because, you know, she doesn't have as many eggs left for the second night?
Yes, so the same females can come out back to the same site. They may try slightly in one night and then try more another night. I haven't actually tracked a single female though to see, you know, energy differences between an individual female. So I'm not quite sure if that is what's happening for individual females.
And not a related question, but you're doing plankton tows. Are you doing it in the middle of the Nassau aggregation and then in the middle of the tiger aggregation so that you know that your eggs are the right species?
Yes, I'm spawning on different nights as well. So on Nassau nights, it's just Nassau, and on tiger nights, it's just tiger.
We have a question from Richard.
So you've had a large increase in the output over the years. Have you seen any, any at all, or an increase in the things that would likely like to feed on that, like basking sharks or manta rays or whatever?
While it hasn't been formally studied, divers that have been going out to the site for the past 20 years have definitely noticed an increase in sharks during aggregating times.
But the kind that would feed on the eggs? Because there's certainly interest in feeding on the adults.
Right. That I'm not quite sure of. I'll have to double check.
Are there any more questions? Quickly, just one quick one up the back there.
Thank you. I would like to know, how do you estimate the population size? Could you tell me a little tiny bit more?
Yes, there's a paper by Lynn Waterhouse that was published in 2020 that goes into all of the population estimates. I'm not sure if I have a lot of time to fully explain it, but she has a couple of models that she's been using based off of different video data that we have, tagging data that we have, et cetera, that has been able to inform those models. And the paper is pretty good and it's open access, so definitely check it out.
Perfect, thank you.
Yeah.
Okay, thank you. We're just on time. Thanks, Janelle. Our next speaker is Elizabeth Caddison, fondly known as Sean. And Elizabeth will talk to us about snappers. Over to you.
Okay, hi, Elizabeth Caddison. I'm from the University of Virgin Islands in St. Thomas, USVI. Today I'm going to talk about a new project that we started working on, which is investigating the potential of using robots and AUVs to monitor existing spawning aggregations and to potentially explore our reefs to find new undiscovered fish spawning areas. Okay, sorry. How do I? Okay, great. Okay, so there's been 3 big changes in the last, 3 decades, 2 to 3 decades that have really changed the way we do ocean exploration and marine science. One of them is the development of digital photography. Changed everything. The way that computers have developed with their speed, their power, data processing, storage has really changed things, their size. And then the development of underwater vehicles, ROVs, AUVs, and their ability to load this equipment on board and go places that human beings have never been able to go before in our oceans. Now with the development of AI, that uses a variety of platforms to do a variety of tasks. And these can now also be loaded onto underwater vehicles. And so potentially, in some of y'all's lifetimes, we may, scuba diving for marine science may become a thing of the past. We may go to, I know, terrible, right? Yeah, right. For today, with coral reef monitoring, it's still primarily diver-based. There are some pros and cons of this. Divers are, they can readily change objectives, direction, this, that, and the other based on changes in their environment. They take precise, very focused observational data, and when they're in sensitive habitats, there's gentle and sensitive handling of the environment. On the other side, there are physical limitations with divers surveying. The most obvious ones are depth and time. It's dangerous. Scuba diving's a dangerous thing to do, especially in exposed deep areas. It's slow, it's expensive, and there's some subjectivity involved in data taken by human divers and human error. Machine surveying gets efficient and accurate data. It's cost effective over time. There are not those limitations on time and depth, or not as many, and it's safe. Nobody dies putting machine down. On the other hand, the initial startup cost is high. It requires specialized training for operation and maintenance, and there's limited maneuverability in very complex environments. So I study spawning areas, spawning aggregation areas. The most well-documented area that we've been studying over the years is the Gramonic Bank. It's about 10 miles south of St. Thomas, 10 kilometers. It's in 30 to 40 meters of water right on the shelf edge. It's a multi-species spawning aggregation. We have 4 species of snapper that spawn there, 4 species of grouper, assorted other pelagic and benthic fishes. We have been studying it for 20 years, mostly with visual diver censuses. We've also used hydroacoustic tracking there. We've used acoustic monitoring to document vocalizations of courting groupers, and some ROV work we have done there. It's been very expensive and very, at times, dangerous. We're interested in introducing robots to the scene to see what they can help us with. Our objectives would include monitoring the fish abundance and size and spatial distribution of spawning fishes, as well as perhaps to video spawning behavior at times and places that we can't get to. And then eventually we'd like to send robots out to collect data on critical habitat and perhaps find undocumented fish spawning areas. So we need a special machine for this, and we have come across the Ranger Bot. The Ranger Bot is a small AUV. It's out of Australia, Queensland University of Technology. It was developed by a guy named Matt Dunbobbin. It's a vision-only AUV. As I said, it's small, it's lightweight, one person can handle it. It's vision-only, its tasks, as well as its navigation, is controlled by 2 pairs of stereo cameras that are mounted on it. It has a depth rating of 100 meters. It has a battery life of 6 hours. It has this great thruster configuration, 3 on each side, tilted at different angles, which can make it incredibly maneuverable. And so the Ranger Bot is capable of real-time perception-based tasks. It is programmed on the surface from a laptop or a tablet, and then is completely autonomous. The navigation, again, is based on these stereo cameras, measuring distance on the bottom. It's also maintained at a certain position off the substrate using stereo cameras. It is equipped with a GPU. So it's, in the last several years, the RangerBot's been used to do really accurate, high-definition seafloor mapping on the Great Barrier Reef, as well as coral reef monitoring and water quality monitoring. And maybe more interesting, excited, the RangerBot has been used to identify the crown of thorn starfish on the Great Barrier Reef, and to not only recognize it and find them, but to swim, fly, whatever, to approach them and inject toxins into them with this proboscis that it sends down. It's been programmed to do that. So that's pretty cool. And then more recently on the Great Barrier Reef and in the Philippines, the RangerBot has been trained to recognize suitable coral restoration habitat, and to hover over these areas and to disperse coral larvae. So it's perception-based tasks, which is what we're looking for. So what we needed to do is develop a model to identify the fish species that we're interested in. We use convoluted neural network platform, which is a deep learning AI platform for object detection. It's well known, it's well used for that. In particular, we used an algorithm called YOLO. This is under the CNN umbrella, and this is used by other modelers who work with fish identification, and it's preferred because it has really fast performance and it does real life processing. So the first thing that we had to do was we had to take a YOLO algorithm and we had to train it for the fish we were interested in. So we collected over 65,000 images of all different fishes, primarily the 6 common snappers we have, the 4 groupers. The groupers we actually collected not only the main normal color phase, but also spawning color phase, courtship color phase, and the optimal number that YOLO likes to do its learning is 1,000 of each object or each fish species, minimal 500. You can see that we're close to 1,000 on some of our fish, but some of them pretty depauperate. But after about 6 months of collecting these images and annotating, we decided to move on with our models. So what we did was we took our images and we uploaded them into CVAT, which is an online annotation program. And what CVAT does is you tell it to delineate or annotate an image from the background, and it will do that, and then you label that. And so you take those images and their annotations, and after we had to take them from pixel images to pixel mass to polygons, another step, but we did finally get them into the YOLO framework. And the YOLO framework took 90% of those images and annotations and trained on them. And then it took the other 10% of the annotations and tested the models that it came up with on those annotations. So and this is an example of what you see when you look at the YOLO program. We came up with 2 models. We have a snapper model where all our grouper species are lumped and our snappers are separated by species. Then we had a grouper model where our groupers are separated and our snappers are all lumped together. And so YOLO tested itself on this model. This is the snapper model. The true snappers species are on the bottom, the x-axis, what was predicted on the y. You can see that with the snapper species, you get between 84 and I think 94% accuracy. So this thing was really good. This model was really good at predicting snapper by species. And also, grouper lumped together, 95% accurate, and the Nassau grouper bicolor 80%, normal phase 84. Now that's really good. Anything over 70% they say is really good. And this was our first run, so we're pretty happy with this. The grouper model, not quite so good. You can see snappers lumped out, 93%, really good. Grouper lumped out, 92%, really good. The color phases for the Nassau, got a little confused with, called a white Nassau normal phase. Still around 65% for all of the images that we tested. So that's pretty good. Now, YOLO also comes out with a confidence value, and that is basically a probability index. So you can see that the x-axis is the confidence level. At 20% confidence on most of its tests, it was still at 80% accuracy. So we think that's good, and we're ready to go on the road with these models. So we have done sea trials with RoboBot in shallow water, works really well. We have taken it out to deep water, the shelf edge, that's where we want to use it. There have been some challenges. We've had some leaks, some machine problems, and O-rings. We think those are easily fixable. The bigger problems are the currents that are causing the RoboBot to go off its program course. Matt has come up with a floaty bot, which is another robot that you can deploy with the RangerBot that is on the surface. It has a hydroacoustic receiver transmitter that can find the position of the RangerBot and actually transmit very simple signals to get it to change its course slightly, depending on if it's been pushed out of the way. So we're really interested in trying that out this season. And then coming up in the coming spawning season, which is starting in December, January, we are going to continue our model training with hopefully more grouper courtship images that we can annotate and train our models on. We want to take our models and put them on board the RangerBot to see if we can do real-time species identification out there on the shelf edge. And then finally, we want to turn perception into action. We want to program the RangerBot to change its behavior based on what it sees, what it visualizes, whether it's turning, stopping, turning to track a fish, following it, filming spawning behavior, or perhaps sizing the fish. So with that, I'd like to thank our funding institution, Blue Point Conservation Science, also Matt from Queensland University, QUT, and he's the brains behind this whole project, and GCFI. Thank you.
Thank you, Sean. And bots and AI are certainly part of our world, and thanks for reminding us that they're taking our jobs. We've got time for a question. We've got several. Probably this one here, please.
I want to say I did forget to acknowledge everyone on the front page, our students and technicians who are the actual annotators and modelers here. Big thanks to those guys because they spent many, many hours getting these models working.
Thank you for your presentation. Amazing. It was so wow. I have a question. How many times did you take to implement the model and train the model to the different species?
I'm sorry, I didn't.
How many times did you take to train the model to identify the different
so
species?
we took, each of our annotations go through the YOLO program 100 times, 100 epochs. That was a recommended number. I don't know that our programs really picked it for a particular reason, but that was the recommended number of times the annotations went through the model.
Thank you. We can squeeze one more question in. Quick, first hand up. Richard. No, microphone.
So how much does a RangerBot cost?
You know, the machine itself is, say, around $30K. You know, it's cheap for an AUV, probably, like that. I don't know. There's also, you know, we've had Matt Dunbob down to train us, to train and program and do a lot of the work, so that was an added cost on this project that has to be taken into consideration. But yeah, about 45 grand.
Thank you, Sean. I think we're going to have a lot more questions, but let's hold them for after the session if that's okay, so we can move on. Thank you. Next we have Martha Prada. Welcome, Martha. Martha's going to take us through the world of WCAFC.
Good morning, everyone. Thank you for being here, and also good morning to people online. Today, I'm going to present results from a regional pilot project that was developed through the WCAFC working group on fish pond aggregations. And so it is under the leadership of the working group convener, Miles Phillips, who is unfortunately couldn't come, and other co-authors, as you can see in this slide. The initiation of the project actually comes out of the formal adoption of the regional fish pond aggregation management plan. This was an initiative from the working group in the past, I will say, 8-10 years, that finally goes through the different process to get formally adopted at the WCAFC 18 session back in 2022. And after that, it was also adopted by the CRFM mechanism and the UNEP Sport RAC 2 as one of the species on their annexes. The funding of this project came from the EU DG MARI project. This pilot project focuses on 3 countries, the Belize, Bahamas, and Cuba. And it was comprised of 3 components. Component 1 was basically aimed to gather information, focuses on the 2 species that the regional management plan is being adopted, the Nassau grouper and the mottle snapper. And so the second component was developed to be able to utilize, compile information, and look at the WCAFC fish information system and how the 2 kind of data sets can be combined, and how that can be put on into more regional management recommendations. And component 3 is focusing on education and outreach, actually one of the very nice products of this project through the Big Fish initiative that produces a digital hub and Big Fish campaign. This project was conducted, it's just about to finish with the reporting. Major outcomes you can see in this slide. On the component 1, there was several stakeholders workshops led by different NGOs and government participations in the pilot countries. And also it got information on the spatial distribution of the spawn aggregation on the 2 species and other aggregating species, and also it tries to compile additional information or publish that is not available in the national reports on landings on these species. So component 2, I won't take a lot of time to describe that because that was presented yesterday afternoon by Nancy Cummins. And I move on the component 3 on the Big Fish initiative. You can see here the QR code and perhaps all of you, or majority of you, are be able to see and look at some of the products, the 1-hour documentary that was presented on PBS, the more than 5 short documentaries, the radio kits that were supported through the Sport Rack, and the hub actually now contains a lot of published and unpublished information on the species for the pilot countries. So with all of that, what lessons we learn? To be able to perhaps have a better understanding on how this spawn aggregation works and can be managed, it requires a lot of more uplandings information. So it requires many different kinds of information, most of them not published, and it's on the traditional knowledge, so illustrate the importance of combining scientific information with traditional knowledge in a way that can be effectively put in place into management regulations. But despite all of this gathering of information, still significant data gaps remains, and that is mostly because majority of the databases are not at the species level. So it's on big gross categories like groupers or snappers, and so it's difficult for do our evaluation on fishery status, stock status, and so other when you don't have information at the species level. With that, I not recognizing that not all of the spawn aggregation sites at those countries are known. Seasonal protection can be an effective management, just in like a precautionary approach. The WCAPC has been calling for seasonal protection again for more than 8 years, and it's been in place in some countries, but not in other countries. So still we have things to do. And even we compromise at regional meetings and workshops, once you are back in your countries and having limited resources, limited capacity of personnel, and dealing with more than 5, 10, 20, sometimes different fisheries, only a handful of people, it becomes difficult. And so some of that information is being recognized that, for instance, in the case of Nassau grouper, which is in critical states at different categories by different organizations, it demands that the national authorities and actions needs to follow up with this. So another lessons learned, that the gathering information from the stakeholder is as important as from scientific sources. And all of those different kind of knowledge has to be combined, and actually came out with streamlined regulations that in a way it can be implemented in some aspects. We cannot continue doing regulations plus another regulations from another species and another compromise if you are not really able to enforce at least a minimum part of that. And in that way, the promotion and the use of digital hub, we are thinking that is a very good way to communicate this message. Communication at the end of the day is really important, and perhaps it is a way to involve everybody. And as long as you feel involved, you can do something even if it's not in a regulation. So we have also identified next steps, and so they are highlighted in this slide. So now it's a pilot project. This pilot project now is going to be following through the WCAPC and other regional organizations' formal process for updating their work plan in the agenda. They perhaps need to do support, technical support, on how these implementations and regulations can be formally introduced. There is one important thing that the Regional Fisheries Management Plan, which I didn't mention at all because it's not the focus of this presentation, it calls for an establishment of a regional coordination group. And that group can be established and provide support in different ways as the country are needed. We had in the previous GCFI meeting an informal workshop with representation, majority of the experts in the region working with spawn aggregation, and there is guidelines to establish that regional coordination committee, which is really a need to follow up and not stated in the report of a pilot project or stated in a regional fisheries management plan. So I won't spend much time in the recommendations, but what I'm saying in these slides is all those learned lessons needs to be incorporated into the formal procedures and become, in a way, available for countries to implement. The importance of having data collected from different sources, I like to highlight that, and I like to highlight the fact that in those countries, Belize, the Bahamas, and Cuba, we were able to work with local NGOs, involve local governments, and that collaboration needs to be stronger and continue through time and expanding to other regions. Dr. Krista was the responsible for the Bahamas case study. She is here, so if you have particular questions on the Belize case, you can feel free to, I compromise on you, Krista, to ask her about the results, the experiences, because I cannot directly transmit all the perhaps emotions, but in a way some frustrations too. It's not easy to work. You need to go through a whole of paperwork, regulations, permits, whatever. So it is not easy, but it can be done, and it needs to be done. We need to move from regulations to actual enforcement. Thank you.
Thank you, Martha. Data, we need lots of it. That's very important, so thanks for that. Open up for, we've got time for some questions. Just raise your hand. No, you presented really well. It was full of information. Richard.
Not so much a question, but a comment. You mentioned that the species-specific data is really a problem, and there's something where AI can really, really help because it can recognize things better than humans if it's trained enough. And so you can even take the human out if they don't know it, the machine will do it for you.
Actually, we are right now in a process to drafting a project for FAO and the EU funding again to try to use AI for this kind of work. So you are a step ahead, Rich.
Another question?
Maybe I'd like to have a comment. Mm-hmm. What I like most, the working in this arena, is the possibility to combine the work that the conservation organizations are doing with the work that fisheries management are doing. If we are not able to combine those two objectives, we won't make any progress. In particularly the case of spawn aggregation, the regional management plan is not focused on fisheries, it's focused on conservation. But we need the fishery associated with that to be able to combine those. And so that's important, a message that I want to leave. Thank you.
Thank you. Okay, we're up to our last presentation today, and it's a presentation that will take us to Key West The speaker is Danielle Morley, but the infamous Alejandro Acosta will be giving the presentation. Welcome, Alejandro.
Sorry, Danny, he cannot be able to be here, but I'm very familiar with this project. I used to work for this agency until April for many years, so I think I can give the talk. No as better as she will do it, but I will do my best. But I want to follow up in the point that Marta just made to close her presentation. Last year, I talked about the role of the NGOs in fisheries, especially in fisheries aggregations and all that. And this presentation here is going to have a lot of that, because this has been a multi and a holistic approach to engage all the stakeholders to do this. So, but this will be the end of the talk. Let me start with this one. So I'm going to be talking to you, and I'm going to bringing you more to a regional in Florida, what is called Western Dry Rocks Science Plan. It was for us an opportunity to develop a science plan to present to the Fisheries Commission in order to try to protect an spawning aggregation site located. Everybody knows how spawning aggregation form and what they do. So we just talk, all the speakers talk about that. But Western Dry Rock is located very close to Key West, and very close to the mainland of the Keys. So it's an area that is accessible to everybody, and for many years was very popular for fishing spawning aggregations, to the point that used to be advertised at the radio from April to July to August. The moon is coming, so let's come to fish. So it was really a very well-known spawning aggregation. So it was not easy to work in this place. But based on the work that NGOs did, working with us together, we were able to document the aggregations and to document the area. And we were able to present it to the commission to see this area needs to be protected. What can we do? Okay? So in 2021, we gave a presentation. We had been working in spawning aggregations in the Florida Keys for 15, 20 years too, and we were able to work in the Tortugas area that was one of the more well-known spawning aggregations in the Keys, but also was protected. So the work was very easy to do because there was already a level of protection. But Western Dry Rock was completely open. So in 2021, we presented our preliminary results of the work done in Western Dry Rocks and the Tortugas to the Commission, and asked for a way to protect that area. And the Commission decided to protect a small square area of 1 mile for 4 months, going from April to July, with a caveat that we had to present results at the 7 years of protection to see, is this area really working? Are we going to keep it? Are we going to let it go? And that was the same thing that happened when we did it in the Tortugas. So to do that, we say, yes, we can do that, but we need to have funding to conduct research and to do monitoring. And there was a commitment, an indirect commitment, and they were going to provide the funding. And so far, it has been like that. We have been providing with the funding to develop the data and all the information that they need to outdate them and to come back to this after these 7 years to report what happened in Western Dry Rocks. It's a multidisciplinary team, and still in these pictures, I should not be there anymore, but I'm the first one here. But we have social scientists, and we have a lot of field work. So it's not really 1 or 2 persons who do this job. It's a lot of people, it's a lot of work. And what was the approach? At the beginning, we have what we call a multidisciplinary approach. We have biological scientists and we have social scientists. And they were, we were trying to start working together and doing that. Then we moved into developing a science plan. Why? We needed to have something that will create a roadmap for us of how to do, or what we wanted to do, how to reach the goals of where we wanted to go, but at the same time, how we can provide accountability to the commission and to the managers of what we are doing was really the way to do it. So that is where we start working together, biological science, social science, with a goal in common. And the end, at the 7 years, is try to have a transdisciplinary approach where we have everybody involved, all the stakeholders. We have the biological, the social, the managers, and the resource users all together to protect the aggregation or sustainability of those resources in Western Dry Rock. So we started to develop the science plan. I haven't seen the final copy, but that's what it looks like. And the goals were two goals mainly. One was focused on the fish resources. We need to do an evaluation and monitoring of who is using that area. It's a very small 1 mile, so this is tiny in comparison to the vast location of the Keys. But the Western Dry Rock, that small piece has been documented and is a multi-species aggregation site for many species, snapper, groupers, grounds, permit, and pelagics and all that. And the second point was because it's so close to mainland and it's an access to fishermen, not only commercial but also recreational, how the users are impacted for us closing this area and protecting these aggregations. So there has to be two fronts that we try to approach and try to get the answer for these two. So the monitoring one, we had used almost all the techniques. We were lucky enough that we have had the funding to go from AUBs to ROBs that we have in place systems to do that. We have a lot of acoustic tagging, we have hydrophones, we have aerial surveys, visual sensors. So when we go there every year from April to July, every full moon is a large amount of data being produced, and it's a lot of being collected. And that's one of the problems we're facing, and that's why we're bringing artificial intelligence, also working with somebody at the University of Queensland, trying to have automatization of how we can identify and count all the fish that are being collected in the videos, on the water cameras, and the data from the hydrophones and all that. And also the ROB is different because we work, the AUB, we work with Florida Atlantic University, and they can provide to us the data or kind of analyze for that. But I think we are all going into the same direction of what Sean was mentioning. Technology is getting more affordable, and I think that is what is going to happen at the end, when we have to monitor these large areas, and for very specific long periods of time, we need to have automatization because people power is really limited, and people get tired very easy. But we use all those techniques. And then we have the socioeconomic component. So one way for us to approach that is conducting interviews, meeting with people, and then we have a group of socioeconomic staff different to the scientists because they know how to approach the fishermen, how to talk to them. Scientists, sometimes we don't have the charisma that Will Heyman has and all those kind of things. So that's what happened. And then the other way that we need to know is how many people are using really the area. So we conduct aerial surveys, and we have underwater hydrophones that will allow us to know how many people is crossing through the protected area or how many boats are stopping there to fish a day and night. It is really a lot of poaching going on. So the hydrophone is providing us right now that information. In the plan, we had designed different matrices how to evaluate the plan, and it's trying to make it as simple as possible. It's like a traffic light, red, yellow, and green, and that was to demonstrate, are we seeing the aggregation numbers increasing, staying the same, or they are decreasing? And the reason why we have to make it simple is because at the end of the day, this is going to go to a commission meeting where all the public is going to be there, and they need to understand in a very easy way how this is evolving and what results are we getting. This is the size of the Western Dry Rocks. There is a movie here. And here I'm going to show you a small movie that we have. Can I click it? Okay. So, and then look at the time, the periods. That's April. In April, we start seeing aggregations of permit. And then in May, we start seeing mutton snappers, crabell jacks, permit, horse, another jack. And you see the AUB and different tools collecting that information. Then by June, July, we start seeing more the mutton snappers and the jacks again. But this is only in this small square, in this mile of protection. And then in July, you see you have a gray snappers and you have other jacks. So that data is giving us the information, the spatial and temporal information for us to present. Yes, the aggregation is taking place. So it has been a long journey. We learned a lot from the previous work that we did. And the most important thing has been to integrate a holistic approach to bring not only science. The science is not, I don't gonna tell you it's cheap. It has been very expensive. You have to deploy a lot of instrument, and not only that, people always think, oh, how expensive is the AUV or the it's the manpower to analyze the data and to go in the field. Every time we go for a full moon, every month, is 7 days, then you have to have 6 people working there all the time. They're long days. So you need to have really an investment in science and in monitoring and a support of people who believe that this is important. And I think if we, after the 7 years, we go to the commission and we present this data, most likely they're gonna give us 20 more years of protection. That was what happened last time we did it in the Tortugas. We went there, we did it, and we were for 7 years, and we asked for 7 more years, and they say, no, we're gonna give you 20, because they saw it's working. So science, biological science, social science, and all that together will really produce results, but it will take time and effort to do it. So that's what I have. I'm sure Danny would have done a better job, but that's what I have. If you have any questions.
Thank you, Alejandro. Yeah, science for management, actually progressing through to getting a management change is so important. We can do lots of science, but if we need change to happen, we need public buy-in and the political buy-in as well, and that's a hard thing to do, so well done. Yeah. We've got time for questions here, so I will open up for first hand in the air. Will?
Thanks, Alejandro, and thanks, Danny, and your whole team. You guys are crushing it. It's really impressive.
Well, I'm not there anymore, so they are crushing it.
Humble guy. No, my question has to do simply with the regulations and the impact of the regulations. I understand it's no take, but is there any sport fishing allowed, and what are the implications there?
There is not really a sport fishing allowed. So that during that time of the year, it's closed for those species. It's completely closed. There may be other species that they can harvest, but not for those species. So there is a protection there for those species.
But originally,
it's a closed area. That area is closed for fishing. Yeah, yeah, it's closed for fishing. Yeah.
And prior to closure, though, it was found by commercial or sports fishers, or how did you find it?
Oh, no, that like I said before, that aggregation has been going on there for commercial fishing and recreational fishing. People from Key West and all over will go specifically there. It was advertised in radio, you know, the aggregation is coming, let's come go fishing, and all those kind of things. So it was very well known.
Got a question up the back?
Ori, you used to work there.
Dorothy did.
I know. I didn't get a picture or anything, though. My question is just, if it was so well known for so long and so fished so hard, do we know of other species that were aggregating there that are not anymore? Groupers, maybe?
The first thing everybody will know, those aggregations for the commercial species. There is a lot of other species like porkfish and grunts and all that, and parrotfishes and all that, and they aggregate there too. And those ones, they don't really get the spotlight like the groupers and the snappers. So it mostly was a mutton snapper aggregation site, because not even the gray snapper can aggregate in large numbers. It's not really targeted like they were targeting the muttons.
But there's no evidence for like Goliath or something?
I don't know. I don't know about that.
We have a question right here.
is there, do you think that the 1 by 1 mile squared area is enough, or is there, do you think, capacity to expand the protected area beyond just extending the lifespan of that 1 mile squared area?
It all will depend probably on the political will and the need of the situation at the moment. When the 7 years end, and they should be ending in 2028, 2027, if the data is strong enough to support and the benefits of closing has been shown throughout the year, most likely that can be proposed. But at the moment, this is the first instance we got and what we were able to get. It's a very political, complex situation. Sean?
Sean? Just get the microphone, sorry, so the interpreters can hear it.
On that note, the periphery of the closed area, is that heavily fished? People get as close as they can and maybe sneak over, and is that a problem?
Yeah, there is no question that there is a spillover, and then there is boats around, and everybody knows what is going on.
And do you have hydroacoustic data that shows that fish go in and out constantly the way we do?
We don't only have a system deployed in that mile, we also go outside, and we have a large visual sensors program that goes all over the keys throughout well, you're familiar with the RBC that we have throughout the whole year, and then we deploy a lot of other monitoring projects and programs that we have that will give us an indication if there is benefiting the spillover is happening, or there is other areas that we're looking all the time. Yeah.
Thank you. Are there any more questions? And actually, this is a good discussion to have. And if there are any further questions for the other speakers, let's use this time. We're actually into lunch break, but I think we can have a bit more of a chat, if that's all right. Put your hand up if you've got a question here.
Hi, Alejandro. Is there any proposal to increase the size? I'm not sure if anyone's asked that before, of the protected area, or is that what you're working to collect the data for now?
At this moment, I don't think there is another proposal. We are in a marine sanctuary, so there should be some other level of protection, and there are zones that they are established and all that, but for that specific area, that's what the size of the area that we have to work, and that's what we need to provide the information, and then from there, perhaps we can get something larger to do that, but this is what will be the baseline for any other decision there.
Thanks, Alejandro. Are there any other comments for the session as a whole, or just for Alejandro?
Okay.
No? Thank you. Thank you to Alejandro. Just a couple more things from me. Just after this session, a few of us who work on spawning aggregations specifically are going to just gather and have a chat. We try to do that every GCFI and share our thoughts and ideas. Another comment I wanted to make is Science and Conservation of Fish Aggregations, fondly known as SURFA, is an NGO that I'm part of. Yvonne Sadovi, Kevin Rhodes, Rick Nemeth and Jan Robinson, we work together on understanding aggregations, but we have a global database on aggregations, got well over 1,000 records in the database. If you're interested in using the database or contributing to it, then come and talk to me. Kevin Rhodes, as we speak, is inputting another 30-odd records into it. So it's a global database on where aggregations are, for what species, when, and habitats and so forth. So it's a big one. The other announcement I wanted to make before you leave is, remember there's a GCFI membership meeting this afternoon. Please come along. It's a membership meeting to give you an understanding of the inner workings of GCFI and a little bit of voting for a previous meeting and our budget, and also some board member changes. So come along, and if you're interested in being part of GCFI, whether you want to be on the board or just helping out, then please raise your hand, let us know, get involved, and never know, you could be up here talking and doing things before you know it. So, and I think that was all. Are there any other questions from anyone? Fadila?
Just specify that the meeting is open to everyone.
Yeah, that GCFI membership meeting is open for everyone who wants to come along. And it's in here, isn't it, Fadila? Yeah. Nancy.
Thank you. I just want to let everybody know that the silent auction is open until lunch on Thursday, and it will close at lunchtime, so go out there and bid. And thanks to everyone who has bid.
One more announcement.
One quick announcement. This afternoon section, the one time start at 2 in the afternoon, is going to be a local Guadalupean section related to blue economic science. It's going to be a very interesting section. The PowerPoints are very good, and it will be really great if everybody can attend and participate, because it's going to be a really good section. At 2. It's in French, but it will have French translation. There should not be 2 parallel sections this afternoon. Oh, the CON workshop is a private workshop for specific people, by invitation only.
And one more thing, if anyone finds a spare bag with a black book in it, please hand it over and I'll give you a reward. At least a drink at the bar.
Recording stopped.